A child sits down to breakfast. She fills a bowl with cereal. The crunchy cereal rings are sweetened with corn syrup. 100% of the corn used to make that syrup comes from plants with DNA that was intentionally altered by crop biotechnologists. She takes a big slurp of corn-sweetened O's from her spoon and splashes some milk on her shorts. The nice new blue shorts that her aunt had just bought her from Target. She grabs a dishcloth and wipes the milk off her shorts, succeeding only in smearing the stain further into the fabric. The cotton in the dishcloth and in her shorts are from plants containing DNA segments originally found in bacteria. That DNA allows the cotton plants to produce tiny crystals, toxic to caterpillars, that could otherwise lay waste to a whole field. 80% of all the cotton produced in the world is from plants whose genetic lineage has been deliberately modified, not just through the gradual but forceful push of selective breeding, but with technologies developed in laboratories in the 1970s and then applied to a wide range of crops from the 1980s to the present day. After cereal, the girl cradles a slice of sweet-smelling papaya that her mother had cut for her. She sits out on the lanai, staring out at the ocean stretching off into the horizon beyond the edge of the city. The papaya isn't from the tree down the street, but from a grocery store, and so it's most likely that the papaya is one of the 85% of the Hawaii papaya crop made of plants whose every cell contains a secret weapon in its DNA. Most of the cultivated papaya plants in Hawaii house a tweaked piece of DNA inside their cells. That piece of DNA comes from the devastating Hawaii papaya ring spot virus. That little template piece triggers the adaptive immune system of the papaya cells, priming them and their antiviral defenses to mark out and destroy any DNA that matches the template piece. The papaya tastes sweet, though not as sweet as mangoes, her favorite. At school lunch, they have spam musubi. She loves the chewy meatiness of the fried spam. It feels to her like a hug. She doesn't think about the pig whose meat went into the spam, or about the genetically modified soybeans and alfalfa that almost certainly went into feeding that pig, growing up its muscle till it's ready for slaughter. The canola oil that the spam was fried in came from genetically modified canola plants. The plants were genetically modified using 3 different species of bacteria. One to act as a carrier to bring new DNA into the plant cells, and the other 2 species of bacteria were harvested for their DNA, each yielding 1 functional unit of DNA each. These DNA units were delivered into the canola plants and allow them to metabolize and break down a herbicide called glyphosate. And that allows the fields to be sprayed with large amounts of herbicide, eradicating weeds, boosting yields, and all without relying on other chemicals that are a lot more toxic to humans. And the company that invested in creating and patenting those genetically modified canola plants just so happens to be one of the major sellers of that very same herbicide the crops are now immune to. The seller nets billions in profit per year out of this whole arrangement. The girl enjoys her spam musubi and heads out to play. Elsewhere in the world, school children might be enjoying genetically modified tomatoes with higher nutrient contents, apples that are modified to reduce bruising, and eggplants that contain the same anti-caterpillar crystals mentioned earlier, the ones that are in the cotton so many of us wear and use. Genetically modified plants are a fact of 21st century life. GM plants are just a small chunk of total cultivated crop species and varieties, but even though GM crop varieties are just a small amount of the total crop species that humans grow, they make up almost 10% of the total global output of plant products in the world. Almost all of the cotton, soybeans, corn, and canola grown and sold worldwide are GM. And a much more diverse array of GM crops have once been sold, will be sold, or are coming onto the market as I write this podcast. Apples, pineapples, potatoes, tomatoes, eggplants, bananas, mustard, summer squash, and not just edible plants, ornamentals too. A genetically modified rose is sold in Japan that produces the blue pigment found in many other flowers, but until the 2000s, never in roses. In America, consumers can buy genetically modified petunias that use the same biological mechanism as fireflies to produce light inside their own cells. In a completely dark room, the plant glows a faint green. Welcome to Modified. I'm your host, Dr. Orlando Delange. Through this podcast, I'll be sharing some of the things I've learned over a decade of work in plant science, and some of my reflections on the way GM crops have shifted the ever-evolving interplay of human technology and nature. I'll try to guide you through the science of genetically modified crops, following a historical journey from the late 19th century and into the near future. Along that journey, we will not ignore the complex social realities, the mechanization and corporatization of agriculture amid global urbanization, the disinformation and the destruction of GM research sites, the moral implications of an expanding ability to modify life's code stack. But for now, at least for the first season of this show, our focus will be on the science. What actually are GMOs? What are crops? What is plant breeding? And how do all of these intersect? We could start our narrative millennia ago, and in one of many places around the world, with people gathering seeds and sowing them deliberately to harvest their products, no longer reliant on the landscape as it's shaped by the thousands of species and powerful physical forces beyond human control. Instead, these first farmers started to push back, and they admired the products of their work, a sign that they could make more and do more than any other creature on the planet. But then why start there, and not way back with the first humans in Africa 100,000 years ago, creating stone and bone tools, fires, ropes, clothes, acts just as revolutionary as farming? And why narrow ourselves to humans when we could look back almost 4 billion years to the last common ancestor of all living things? Scientists called that cell LUCA. LUCA was in the vanguard of a new type of matter that organized itself, that harnessed chemical reactions to build more copies of itself, fundamentally reorganizing the energy landscape of our planet. Modifying nature is as old as life itself. But no, as much as I would love to spend hours and hours rambling on about the history of life on Earth, we're going to begin in the much nearer past, in a world that doesn't seem so distant or so different, though we will be in a castle, more specifically in the castle kitchen, in a university town in Germany, where a scientist called Friedrich Miescher is about to be the first person to purify DNA. The story of Friedrich Miescher. A common theme that you can find in the life stories of famous scientists, particularly from the 19th century and still today, is that they were not the first scientist in their family. As with really anything in life, exposure and opportunity can profoundly shape motivations and abilities. And so it is for Friedrich Miescher, who was born in 1844 in Basel, Switzerland, into a family with multiple successful biologists and physicians. Another thing that shaped his scientific career was his disability. He was partially deaf due to a bout of typhus as a child. That led him to pursue research science instead of medicine. Research science was at that time a relatively solitary profession, at least compared to medicine. And I wanted to call attention to this, not because it has anything to do with GMOs, but because unfortunately, people with disabilities today are all too often excluded from laboratory science, so it's worth pointing out how people with disabilities have always been, and continue to be, part of the scientific community. He completed medical school to please his father, but then immediately found a research position in Tübingen in Germany. And that just so happens to be where I did a good chunk of my own PhD research. Though whereas I was working in the then shiny and new Center for Plant Genetics, he was working atop a neighboring hillside in the kitchen of the medieval castle of Tübingen. Even though he was working in a castle kitchen, it had been set up as a laboratory with a good amount of well-made equipment and an enviable chemistry set that Miescher could use to investigate the chemistry of a then little understood part of the cell, the nucleus. Miescher, then in his early 20s, was swept up in what he saw as the cutting edge of biological science, the chemical analysis of cells. In earlier decades of the 19th century, scientists studying tissues using microscopes had been able to show that all living things are made of cells, and that cells are hugely complex, diverse, and dynamic structures. The challenge facing biologists in the 1860s is, I think, the same basic challenge that underpins biology as a science. How to find within the chaotic diversity that is life some common building blocks that connect it all. Those building blocks could be theoretical. They could be principles, such as the principle that all life emerges from other life. And by the 1860s, it had been definitively shown that all cells can only arise from other cells. Or could be the principle that when new life emerges, it is similar to what it came from, i.e., that traits are inherited. We call this principle heredity. Or those building blocks that biologists hunt for could be physical things with specific chemical properties. Misha was driven to connect both of these, the theoretical and the material. He wanted to understand the specific chemical properties of cells and to understand how information is passed down between generations, producing the phenomenon of heredity. It would take about another century to crack that challenge. And scientists are still working hard on understanding all the nuances of how information is encoded into the chemical units and interactions of cells. But Misha made another important early contribution. The late 19th century was an exciting time for biologists. There was a flurry of discovery and activity, the product of multiple lines of study and technology coming together. There were advances in scientific instrumentation, such as the improved microscopes that had been made possible by centuries of study into the physics of light, as well as improved manufacturing processes for lenses. There was also a market of people wanting to buy microscopes. There were more people in general, with global populations starting to shoot up, and the people were more connected. Travel and communication technologies were allowing information, including scientific research findings, to be easily and quickly shared between scientists working all around the world. And so although Misha worked mostly alone in the basement kitchen of a castle that sits alone on a hilltop, he was aware of the thousands of other scientists that were working on similar challenges all across the world. If I were to make a guess, I think that more than just curiosity, he was driven to prove himself in that global scientific community, to establish his worth, and to use his scientific skills as tools to help him forge new relationships. We know at least that he corresponded frequently and with a relatively stable set of mentors and other colleagues. Nevertheless, it does seem that pure curiosity was also a powerful driver for Misha, like it is for many scientists, and many people in general, whatever it is that's pulling them on and holding their attention and fascination. He dedicated his life to his work, even to the detriment of his health, and would spend his vacations working in the lab. To the extent that Friedrich Misha is remembered today, and he's not very well remembered, it is because he was a first to do something that we now, through hindsight, know has changed the world. That first was the isolation of DNA. More precisely, what he isolated was what he called nuclein, an acid preparation from cells that was made of some unknown type of molecule. His starting material for this work sounds unappealing, but was very carefully chosen: human pus, specifically pus from fresh bandages. They had to be fresh so that there was no bacteria or mold to contaminate the results, and also so that the human cells on the bandage was still relatively intact. Pus itself is a good starting material for a few reasons. One, it is a fluid. This may seem a small detail, but if you've worked in a biology lab, then you know that fluids are much easier to work with than solids. They're easier to filter and analyze, and what Misha wanted to filter out and analyze was the specific cells in pus, the other reason they're a good starting material. What Misha wanted to filter out and analyze was the specific cells in pus, which is the other reason it's a great material. Pus is full of white blood cells, or leukocytes. Not only are white blood cells freely suspended in pus and therefore relatively easy to purify, but they also have very large nuclei, larger than most other cells. And since Misha wanted to understand the chemical composition of nuclei, they were a great choice. Later on in his career, he worked with sperm cells for similar reasons. They are freely suspended in fluid, not embedded into dense tissues like muscle, skin, or bone, and sperm cells, like white blood cells, have large nuclei. In particular, he used salmon sperm, which are still to this day used when you want to extract large amounts of DNA for use in research experiments. Like all scientists working in an area where little is already known, he failed a lot. He spent months on projects that proved fruitless, and he spent a lot of time getting distracted from what, in hindsight, seemed like the most important questions and challenges. But he did still achieve some major breakthroughs that enabled the field that we know today as molecular genetics. Those were a protocol or method for isolating nuclein, aka DNA, and a description of the major chemical properties of nuclein. Those products enabled others to build on his work and dig deeper, understanding ever more about this mysterious substance. He was not able to prove the nuclein was the hereditary material, that stuff behind the phenomenon of heredity. And he did not prove that nuclein was the physical substance that carries what Austrian scientist Gregor Mendel referred to as factors, and what we know today as genes. Mendel published his pioneering research into inheritance about the same time as Misha was working away in the kitchen laboratory in Tübingen Castle. However, it is unlikely that Misha was aware of Mendel's research, but he was aware of the general debate that was happening among biologists at the time about the molecular basis of inheritance. Mendel's work, which we'll explore in a bit more detail in episode 3, revealed some of the startling predictable ways that traits are inherited. But it has always been known that plants and animals can inherit traits from their ancestors. It is an obvious feature of life. Children look like their parents, whether those are human children and their parents or pea plants and their offspring. And biologists wanted to know how this was possible. What physical thing, most likely something tiny locked inside cells, made this possible? What is the hereditary material? Today we know that the hereditary material is DNA. But back in the 1860s, the scientists working on this problem only knew that they were looking for some sort of molecule that was very complex and diverse. It had to be complex, so that it had lots of parts that perhaps could be arranged in unique ways as a form of information storage. And it had to be diverse, so that you could have different versions of the molecule carrying information about different traits underlying the diversity of life on Earth. The number one suspect, and one that would remain the top suspect for decades more after Misha's work, was proteins. You'll learn more and more about proteins if you stick with this podcast. They are the workers of the cell, carrying out a huge variety of functions, with different types of protein carrying out different tasks. They are also physical building blocks, with many biological materials, from muscle fibers to the keratin of your fingernails, made from the same type of protein packed into bundles. In the 1860s, there was still a lot of things unknown about proteins, but some of this incredible complexity and diversity was already known, and it made proteins the prime suspect in the hunt for the hereditary material. Misha did consider whether nuclein might be the hereditary material. It seemed to be one of the main things inside nuclei, and studies of dividing cells showed that the nucleus is a key player in cell division, and so may hold the key to how information is passed on down generations of cells and down from parents to offspring. However, when he carried out a detailed chemical analysis of nuclein, he found only that it seemed to be made of carbon, nitrogen, oxygen, and phosphate in similar amounts, no matter which cells you extracted it from. He showed that it would react to acids by coming out of solution and becoming a solid, and that was crucial to the isolation protocol. But other than this, nuclein didn't seem to do anything very much. Unlike proteins, nuclein didn't seem to drive any particular chemical reactions if you mix it with other substances. It seemed altogether pretty boring. Nothing he could point to that suggested that nuclein hold the diversity and complexity necessary to be the hereditary material. Despite giving up on nuclein as the hereditary material, Misha did hit on an idea that was surprisingly close to the truth of how DNA encodes information. And if you are not sure how DNA does that, you'll find out in episode 2. But going back to Misha, he didn't think that his nuclein was the secret necessarily, but he thought that perhaps even seemingly simple chemicals could hold a wealth of information in their stereochemistry. Stereochemistry is the orientation of atoms in a molecule. You can take the same set of atoms, arranged in basically the same way, say a ring of carbon molecules, and in one case there is an oxygen atom and a nitrogen atom, and both stick above the ring, but in another case the oxygen sticks out above the ring, and the nitrogen sticks below the ring. This orientation in space matters, and is in fact closely controlled in biology. When chemical reactions happen outside of cells, you find that you get a whole mixture of different stereoisomers, versions of the same molecule with different orientations of atoms. But when chemical reactions happen in cells, there is almost always a very strong preference for particular stereoisomers in both the inputs and the outputs to the reaction. Not that all of this was fully understood when Misha was working, but some of it was, and so Misha considered that perhaps hidden within seemingly simple molecules, a complex and diverse world of stereoisomers might be hiding the code of life, specifically the stereochemistry of proteins. He was wrong that the stereoisomers of proteins are the hereditary material, but he was actually right that complexity and diversity can be hidden inside seemingly uniform and relatively simple molecules. The very DNA, or nuclein, he isolated. In the coming decades, researchers determined that DNA is long chains that are very repetitive, made of only 4 slightly different subunits that are called adenine, guanine, cytosine, and thymine, A, G, C, and T. In a given nucleus, we will find roughly equal amounts of all 4, but in a specific DNA chain, the ratios won't be equal, and what's far more, the order of these A's, G's, C's, and T's is not random, it is a carefully protected sequence, bundled up and shielded from the most chemically dangerous part of the cell, and monitored constantly by spell-checking proteins that can detect and correct any random errors in the sequence that appear. These layers of protection are so crucial because DNA is that special substance that Misha and many others were driven to find, the hereditary material, the physical thing that carries the information of what makes up life itself. And so we'll leave Misha there, and in the next episode we'll pick up with Gregor Mendel. And in the process we'll leave behind the world of Misha, a world of chemistry equipment, pus-filled bandages, and the messy physicality of chemistry and biology. Gregor Mendel lived in the rarefied world of mathematics. Well, mathematics and pea plants. However, I can't leave off without first clarifying how all this relates to another key piece of 19th century biological discovery, Charles Darwin and the theory of evolution by natural selection. Less than a decade before Misha, Charles Darwin published his most famous work, On the Origin of Species, that found an instant and passionate following among scientists, though also much more broadly reshaping how all of us think about change and progress. While Misha wasn't aware of Mendel's work, he was aware of Darwin. Darwin's work was pretty much an overnight sensation around the world, and definitely around the scientific world. Darwin drew on decades of painstaking observation and experiment to pull together a big idea, maybe the biggest one in biology. The idea that the mixture of heredity and random variation will create change with organisms inexorably driven towards being better and better at surviving in their environment. The same basic principle had for millennia been exploited by agriculturalists to shape crops and farm animals, sifting through the random variation to pick the best individuals, breed them, and see how, over generations, the plants or animals change into versions that are better and better for what the humans at the center of it want to get out of them. And we call this process selective breeding. By the time the 19th century was finished, the work of scientists like Misha, Darwin, and Mendel, and the work of many others, would have spawned a whole new scientific discipline, genetics. And it's one that fundamentally transformed the ancient practice of selective breeding into something that could be done with astonishing speed and precision. I mentioned earlier that I worked in Tübingen for several years, at a center devoted to plant genetics. Much of the work we did addressed fairly fundamental questions about plant science, but we were well aware that the funding for our work and for the Institute as a whole would never have come if it was not for the economic impact of that work, for how it could advance the selective breeding of crop plants. Science drives business, drives science. During my time in Tübingen, I would often carry out hundreds of DNA isolations in a week, but none with the fundamental impact of Misha's work. Even if he didn't know it at the time, he had isolated the substance that carries functional information inside every cell and down generations indefinitely into the future. And he certainly would not have predicted that just over a century after his discovery, that in labs not so very different from his own, scientists would be able to modify and recombine that substance, that DNA. Misha laid the scientific foundations of genetically modified organisms. And if you want to know more about what happened next over the intervening century, make sure to subscribe. And so, thank you for listening to this first episode of Modified. This episode was written by me, Orlando Delange. I used many sources to help me research this episode. I want to particularly mention Friedrich Miescher and the Discovery of DNA, a 2005 article by Ralph Dahm. I also want to acknowledge Pixabay creators for the music used in this podcast, the aptly named Gregor Quendel and also Hugh Parr Pro for the music I adapted for the intro and outro. Since this is the first episode, please, if you enjoyed it, leave a review and tell a friend. And if you have notes, well, I'd love to hear them. You can find out more about me and about the podcast at modifiedpod.com. That's modified, P-O-D, all one word, .com. And you can email me at orlando@modifiedpod.com. That's ORLANDO @modifiedpod.com. I would love to hear from you. I'm a teacher, so I know that if you want to retain information, then repetition is your friend. So at the end of this and every episode, I will leave you with 5 take-home messages, facts or key concepts. If you're not all that bothered about remembering the details, and you're just looking for some casual edutainment, do feel free to skip. The same goes for mini quiz episodes that I'll do a few of as we go. So if you're stopping here, thanks for listening. Until next time. For those sticking around, here are your 5 take-home messages. Number 1. GMO crops are crop plants that have been developed using laboratory techniques to directly modify their DNA. Number 2. There are many varieties of crop plants grown and consumed or used around the world. Most varieties are not GMO. But by total volume, GMO crops make up about 10% of total global crop production. That is because there are a few GMO crops grown in very large quantity, notably various kinds of GMO corn. Number 3. Working directly with DNA generally requires laboratories with a lot of specialist equipment. Number 4. DNA was first isolated in the 1860s, but at the time its biological function was unknown. Number 5. Heredity is the phenomenon of traits passing down generations. DNA is the hereditary material. Okay, that about wraps up our first episode. I hope you'll listen in for episode 2, What Genes Are Made Of. Bye!